Dilution is a process in which you create less concentrated solutions from a stock solution, which is a solution you can use to prepare other solutions of lesser concentrations. First, we transferred some of the stock solution into another vessel for pipetting. For our first dilution, we're going to take 8 milliliters of our stock solution and transfer it to our volumetric flask. This is a close-up of how the 8 milliliter stock solution looks in the pipette. To dilute our stock solution and have a total of 10 milliliters in our volumetric flask, we're going to transfer 2 milliliters of the blue solution or the water solution into the volumetric flask with the stock solution. For our second dilution, we're going to be taking 6 milliliters of the stock solution and transferring it to the volumetric flask. After that, we have to transfer 4 milliliters of the blue water solution into the volumetric flask. For our third dilution, we're going to take 4 milliliters of our stock solution and put it in the volumetric flask. The curve in the surface of the water is called the meniscus. A meniscus forms due to the polarity of water. Glass molecules are also polar. Polar molecules like to stick together. That's why the water in the glass tube is going to stick to the sides of the tube. This is a close-up of how the pipette should look for the third dilution. You should read the meniscus from the bottom of the meniscus curve at eye level. Next, we add 6 milliliters of our blue water solution to the 4 milliliters of the stock solution. We're going to use the results of our third dilution to explain the dilution equation. C1 times V1 equals C2 times V2. C1 represents the concentration of our stock solution. V1 represents the volume of our stock solution. C2 represents the concentration of our diluted solution, and V2 represents the volume of our diluted solution. The concentration of our stock solution was 20%, 0.20, times 4 milliliters, equals the concentration of our diluted solution, which we're trying to figure out, times V2, which is the volume of the diluted solution, 10 milliliters. Solving the equation results in 0.08 times 100 equals 8%. That's the concentration of our diluted solution. Here's a better explanation of how we rearrange the equation to find the concentration of the diluted solution. C1 times V1 over V2 equals C2. 0.20 times 4 milliliters over 10 milliliters equals C2. This results in 0.08 times 100 equals 8%.